All right, this is gonna go quick today because it's mostly some bit of housekeeping for clarifying some of the things we had in the discussion about LFPs and what contributes to them, the geometric influence.
What does that mean again? I heard it was something field potential.
Yes, local field potential.
Okay.
So you stick an electrode somewhere in the extracellular medium and you measure the voltage.
Yeah.
That is a local field potential.
Okay.
If you do that like on a bigger surface area, it's called an EEG. If you do that inside the scalp, it's called an ECOG, or intracranial, EEG of some sort.
Electrocorticogram.
Right, exactly. Oh, okay, sorry.
But there's no GFP. Local field potential.
There's no, well, I don't know. If you record like the whole brain, I don't know how you would do that. Anyways, so the point was just to quickly break down our understanding of that, and can we switch to the other paper, which is going to be the most important one. So I went through this review by Bujaki, which is probably the most authoritative review on LFPs, given that it's been cited over 2,000 times, 2,300 citations or something like that. It's nuts.
2012.
Yeah.
That won't be up to date though. That's just
That's not up to date.
Okay.
Yeah. It's now up to like 2,200 citations.
Okay.
Anyways, just saying this is an authoritative thing. So to quickly remind everybody, there's obviously lots of different influences on the local field potential. So we just quickly want to go through of them and explain a little bit what geometry has to do with it, and want to think a little bit about theta maybe, given that that was like a recent topic. So the first one is quite obviously synaptic transmembrane currents. So this idea that when you have synapses, there will be currents. So for example, excitatory upper NMDA channels open, and so you have a sodium ion influx. So for whenever there are synaptic inputs, whether they make the neuron fire or not, right? Most of the time they obviously don't, you will deplete the extracellular medium of sodium ions, maybe also of calcium ions. And that means that whenever you excite, whenever you activate a synapse, you're going to have a negative local field potential outside of the neuron, which should be pretty obvious. The interesting thing, and that already goes towards the spatial nature of much of this, if I draw like a little pyramidal neuron here with some apical dendrite and maybe some distal dendrites, and the axon out, is that the synapses are not symmetrically placed around all of the neurons. They are local. So for example, all these inputs here that draw up sodium from the extracellular material, right? They are clustered far away from the rest of the cell, but the entire cell is electrically connected via its membrane, which means that this uptake is going to be local, but the field effect, right, of this is spread throughout the neuron, meaning that for electroneutrality, you will get sort of negativity here, but then the return current has to come from all over the membrane, which means you get a dipole of sorts. We will revisit that when we talk about the electrophysiology. The first point is just to note that a big impact on the local field potential is not necessarily activity whatsoever, it's just that there are synaptic inputs, because synaptic inputs move charges.
Well, that is activity. It's just a synaptic activity.
It's synaptic activity.
It's not
right.
action potential. It's not
Right.
sodium spikes, which
Yes.
is one of the first things that's surprising me. Right, to the next point here is they make the point that sodium spikes, which are the thing we all talk about all the time, actually have very little effect on LMP.
Right.
It's mostly, which was a novel idea to me. I didn't know that. It was like, really? So when you think about LP, it's not reflecting the neuronal spiking, but it's not. It's reflecting mostly synaptic inputs, and it may not reflect from the spiking of the neurons.
Right.
That was a
So
big- that was
is
a
this
big
an
one.
order of impact? Just 1, 2, 3, 4?
No, because
exactly.
they argue that the sodium spikes have very little impact.
Can we go to figure 2a? I think it helps with talking about this.
I found the figures in this paper hard. Unusually, the language is easier. So I kept looking at the
I
figures.
think I can
like,
explain most of them.
I can explain them too. I'm just saying interesting that I found the prose quite easy to read. And the figures took time.
Yeah, most of these figures also come actually from other papers. Given that this is a review paper, I guess, I found one of the other papers that I wanted to show is actually where one of those figures came from, but we'll get to that. So just to quickly illustrate that point that I made from above, you see on the top left there a neuron, and it's getting excited somewhere near the apical dendrite. And this is a simulated model, but the good thing is our simulations are very, very good for this. Like the forward properties of the local field potential are perfectly understood, or very near perfectly understood. It's not really much of a mystery, so we can trust these simulation models. And what you see there with the lines is essentially, you see the drop-off in the extracellular fluid near the excitation at that blue dot there, where we stimulate in the apical dendrite. No, that's yeah. Focus on the left. The right one is frequency response.
The line is reflecting that point on the
Right. And so you see those field lines, which essentially show you where the amplitude flips from majority negative to majority positive. So you see that while the effect of the input is local in terms of making the extracellular material so negative, depleting it of sodium ions, right? You have, of course, a return current over the entire neuron because it's internally connected. So the amplitude closer to the soma is actually the other way around. Which is not to be confused with the fact that this is still an excitatory current. So if you were to record the intracellular membrane potential, of course, an excitation at a synapse will have a positive effect throughout the cell internally, but the field effect is different because you have an extracellular ionic flux. So I hope that is reasonably clear. I think we can skip the frequency part on the right. It's simply just the notion that that the frequency response of this is there's a low pass filter to this, so the high frequencies are only measurable close to the dendrite.
Just pointing out that's not skipping it.
Okay. Right. I didn't. Right.
I think you can skip the rest of this diagram.
More importantly, figure 2C, which is sort of the opposite of this. This is inhibition. So, and in this case, they don't use stimulating current, but they simply make basket cells fire near a pyramidal neuron in hippocampus. And what you see is that because, as you probably know, these basket cells tend to provide perisomatic inhibition, so like very close to the soma, and when you record the field potential across the depth of this neuron, so you can imagine a long needle with lots of electrodes along its depth passing by the neuron, then you record different local field potentials depending on the depth. And what you see is that the extracellular medium goes positive in its amplitude close to where the inhibition then actually occurs because of the basket cell innervating that pyramidal cell near its soma. And then when you go far enough out, that amplitude switches. So like near the apical dendrites, the field effect is actually not positive, but negative. That is also shown by this blue line, which is normalized there around the central axis at 0. And so the stuff that is close to the site where you inhibit sees a positive deflection of the field potential, whereas far away you see the return current because, of course, the neuron is internally connected and somehow the charge needs to come from somewhere. So that is just to show that these neurons form poles, right? Dipoles, quadrupoles, and so the geometry is important, particularly when it comes to this effect of the synaptic transmembrane currents.
I'm going to try to speed things up here.
Yes.
That's like I always do, because I don't think there's a lot to be discussed here that's important.
Okay.
Just one thing, going back to the sodium spikes we mentioned earlier, they have a very large effect, but the reason they say it doesn't show up in LP is because it's short.
Yes.
It's like less than 2 milliseconds. So it's not like an action potential doesn't make a big effect. It's just short, and when you're averaging out these things, it doesn't work very well. The other conclusion, I'm just jumping ahead here. Another conclusion is this whole dipole thing doesn't seem to be very important. In terms of usefulness for our work, I'm going to see if you disagree with that. They point out, okay, pyramidal cells will have this, stellate cells will not, because this dipole effect here has to do more with the, in fact, you've got this apical dendrite up there. But they point out that in different animals, it has different effects. So in going from rats to mice to rats to primates, humans, and so on, the dipole effect has more effect and then less in humans. And the reason is because when you line up a bunch of pyramidal neurons, if they're all lined up on the same level, they will add. But if they're at different levels, they kind of cancel out. So the bottom of one is near the top of the other, and so on. So you see very little of this dipole effect in primates.
Right.
It's seen in mice mostly, then rats. So functionally, it's just like saying, well, how would we use this? Does it really make a difference in our work? I don't think so. It didn't seem like it.
Right. So the question is, of course, why would we care about the LFP anyways? And one of the reasons why we got talking about the LFP was because of its signature effect in the whole navigation system. So the fact that the whole hippocampal entorhinal cortex loop has like dominant theta. And you know that plays some role, and so the question was, do we need to properly understand that? And that's how we originally got into this whole discussion.
Yeah. So I don't know if my interpretation was correct or not, but after I read that, I said, no, I don't think it's that important.
Right. The question is, does it do anything functionally? And some people are arguing, for example, that effective coupling, like field effects, play a role in synchronization of oscillations and whatnot. But there's no conclusive proof of that. There's no model that makes
it was interesting. If it went the other way around, if it went from simple mammals, you don't see it much, but you really see a strong effect in primates and humans. And one would go, there's something special there. But it's going the opposite direction, which implies that it's not really useful.
Right. Cool. So let's maybe we skip to the other paper.
Can I just point out one other thing I thought was interesting in this paper?
Yes.
And this was something that struck me as that it might be useful, and I didn't quite understand it all. I'm looking at page 5. I'm not on this one, but in the one you sent out, which was the author version. It's in this section called Spike After Hyperutilizations and Downstate.
Yes.
And towards the middle bottom of that, there's a sentence that says the temporally coordinated silent downstate of nearby neurons associated with positive infragranular layers and a negative in the supragranular layer. So they're basically saying there is an effect here of opposite polarity in the upper layers and the lower layers of cortex. And that it sounded like that was a persistent effect. It's there all the time. And it's sort of these up and down states. So it's almost like they're talking about up and down states. That's what the after hyperpolarization word is. It's a long word for something that we have other names for. So this idea that there'd be alternating positive and negative effects on the upper and lower layers was like, that's interesting. Maybe that's something that's useful. Maybe there's different cycling that, we're doing firing up here, then we're firing down here, and we're firing up here and firing down here. So the down states alternate between the upper and lower layers. That struck me as maybe useful. Okay, that's it.
Right. Can we skip to the other paper?
Yeah, I just want to make sure that I thought that was the most interesting thing I got out of that paper.
Right. If you scroll down a little bit, there's a figure, probably like third, fourth page. Oh, this is already interesting. Just to quickly look at it. So the fact that that the theta reverses its polarity, reverses its phase. There's a full phase reversal along the depth, right, of this electrode. So this is just a rat CA1 probe. So you have all these different electrode sites along that depth, and you see that the field switches around.
Does it always do that?
That is a pretty reliable finding, yes. And that has to do with the geometry of this. And there's models that have been proposed about how theta comes about. Can you scroll a little bit down?
Is that picture showing the actual probe on the left?
Yes, that is showing the actual probe, yes.
That probe is not going orthogonal to the depth there. It's going askew, especially on the bottom part. Slight skew, yes. Well, it's more than slight. If you look at between 10 and 14 or something like that, it's pretty strong. That's a little worrisome interpreting that, because you might be seeing the shift going horizontally. And so like moving across columns in the cortex or something like that.
Right.
So I guess you've already said that this is a reliable effect. But
Yeah, we'll
that
show
little
some figures that
that
shows
picture
that.
there doesn't make me feel confident in that. That alone would explain that, because that could be explained by a horizontal shift.
Right.
So if you were to average all of these guys out, you wouldn't see much of a theta rhythm?
That is a good question. No, actually, it's been shown that so that goes to the models that are used to try to explain theta, which is a dual driver model. If we can scroll down a little bit, and actually they're not perfectly in phase, so you will not get a nice null incline. So this was the little thing that got mentioned also by this person who commented on YouTube. So that this Broca's area, what's it called, the MSDBB, is the medial septal diagonal band, like near Broca's area, where there's so-called pacemaker neurons. I say that in quotation marks because that area is still highly contested. But the idea is that you can nicely explain this reversal of the polarity if you assume that the pyramidal neurons at CA1 are driven by two different oscillators, one that drives inhibitory at the source, so close to the soma, and one that is down at the dendrites there, that is an excitatory source through the perforant path from entorhinal cortex. And what you would get in that kind of a model then is you would get an inversion of the phase along the depth of this, and you will get very nice, reliable theta synchronization. They in part sort of refute this classical model of extracellular theta, or like at least they point out all the problems with it, because there are problems with it, which is also why I said in regard to, I think it was your question on Slack, right? So that this thing is still debated and not entirely sure, and the whole concept of pacemakers makes me personally very uncomfortable because the brain doesn't have
let's just bump up a layer. This is one proposed model. You're questioning it. Other people questioning it.
Right.
Let's start with the big picture. Is there any agreed upon explanation, or are there multiple explanations that are questioned?
Different proposals have been made. Lots of different mechanisms for generating theta exist, and
have any of those settled in? Like that's everyone thinks.
Not that I am aware of. And I think one of the problems is that people want a unified model, whereas in fact the brain is messy and generates theta by different mechanisms in different areas.
Is that your assessment, or is that true?
That is my idea of it, my conceptualization of it, yes.
Okay.
In part because I already showed,
idea then. So yeah.
like we already mentioned, right, that many of these neurons in medial entorhinal cortex have these IH and IT current, which allow them to intrinsically oscillate. Maybe we can scroll up to the next figure.
That was in the previous paper, you didn't talk about that.
And so you don't need, next one after that, I think it's figure 4 or 5. Well, there you show the current sinks, but still further. Here we go, right? So this is an introdendritic theta oscillation. You use QX314 to essentially prevent the neuron from firing, and then you stepwise inject current to change the holding potential of a dendrite. And what you see is that once you pass, some 50 millivolts, minus 50 millivolts or something, you see these very strong theta oscillations, which become very dominant. The arrows there mark sort of calcium spikes that are in there, and they become very obvious in that lower figure B, where you don't block the neuron.
So can we stop for a second?
Yes.
So this was in the previous paper too, and we didn't talk about it.
Yes.
And there was a couple of references in there that I thought I wanted to read, but I didn't get to. And it was under these intrinsic currents. And it was saying that, says only under certain conditions do you get this.
Hold on, what exactly, where are you?
It was the paragraph labeled Intrinsic Currents and Resonances, page 4 in my own. It's the one that introduces IH and IT currents.
Mm-hmm. Right, okay.
And this is where it's talking about, in the middle of that paragraph, there's a sentence that says, when intracellular depolarization is sufficiently strong, the resonant property of the membrane can give way to a self-sustained oscillation of the voltage, and oscillations at theta frequency have been described.
Right, that is exactly what you saw in the
I know. So the question it was in this previous paper. So the question I was sort of curious about this, and I needed to read more about this.
Mm-hmm.
And it always said parasympathetic inhibitory neurons have a preferred resonance and gamma frequency.
Right.
It wasn't clear if these occur naturally or under these experimental conditions where they said you have to have sufficient, they, as you were saying, they were sort of manipulating the neurons to get these things to do this.
Mm-hmm.
So I needed to read these papers, but it wasn't clear that this is something that's observed all the time in naturally occurring neurons, or it's like, no, we have this
Right.
neuron, we can get this to happen.
Yeah, there's one thing one should point out where there's a difference between the idea that you get an intrinsic oscillation from this and the fact that you just get resonance, which is a different phenomenon.
Make sure I understand. Resonance would be something that might die out. Is that right? Or how do you interpret that?
No, it means that you get a neuron that responds by an order of magnitude, maybe better when the inputs come at specific frequencies. So
that's what I'm saying. So resonance is not a self-sustaining oscillation.
but it could lead to one.
Well, that was the big question, I guess. The big question was, there was a big distinction whether do these things actually oscillate on their own, or do they just have this resonance, which is like a preferred frequency? So if you bang them, they'll eventually die out. But you can keep them going by keeping pumping them, right?
Mm-hmm.
So that wasn't clear to me whether neurons actually create these oscillations, self-sustaining oscillations, because they say it can give way to self-sustained oscillations, but under certain conditions,
right.
Or is it more just a resonant frequency of a dendrite, and therefore if you hit it at that frequency, you'll get better responses. So do you know the answer to that question of whether these are self-sustained oscillations?
Well, I mean, these things are always complicated in vivo, but what is well known, for example, is that when you change these IH currents, which is known that they are more strong, along the dorsoventral gradient of entorhinal cortex, where the grid scales and the theta changes, you can show that these recorded subthreshold membrane oscillations change in frequency. And they change in frequency in proportion to the grid, which makes that a hypothesis for the grid scale.
Okay, so it seems to me the more likely scenario, and just making knowing little I know about neurons, the more likely scenario is that there's a resonant frequency to a pipe like a dendrite.
Mm-hmm.
And if you have a bunch of cells that are firing and connect to each other, they will more likely end up firing at that resonant frequencies, because anyways a little bit off, it moves it up, and then moves it a bit later, moves it back, and so they'll just end up doing that. And so the theta oscillation then would come about because of neurons would naturally try to fire together in that resonance, as opposed to, it seems less likely that there'd be a self-sustaining oscillation in the dendrite.
Right.
So I'm going to go with that, unless you tell me that you know otherwise.
No, I think it's good. I think it's fine.
Okay. There was talk about these self-sustaining oscillations, and I think that would be, I never heard of that before.
Right.
So that struck me as like, oh, that's
that's
really true.
I am
that's
not
really
aware of in vivo patch clamp recordings from different dendritic segments to show that there are in fact corresponding oscillations.
Okay, so let's just go on that. I'm going to go on that assumption that it's just a resonant frequency, and that these theta oscillations would arise sort of naturally out of any population of neurons that are basically firing together. Which is, I think, a really interesting idea, because it never occurred to me before that's the way it would happen.
Right, and the interesting thing is also that can be a local thing, like to a dendrite, right? It's not. I mean, it can influence wholesale firing, but it doesn't need to.
Well, yeah, but obviously it's gonna impact wholesale firing, because I'm going to see this theta frequency over a broad area of neurons.
Right.
And if I'm doing an LFP, and each little dendrite segment has got its own little resonance, and they're not in sync, I'm not going to see it. So I think it's an interesting idea. And what's also interesting, because we're talking about theta as a way of encoding, like speeding up and slowing it down as a way of encoding motion, right?
Mm-hmm.
So I think I haven't thought it through it yet, but there's a clue there, because as you point out, so you got a bunch of these cells, they naturally want to oscillate together. And if a cell fires, again, if a cell fires a little bit early, it's like they're all going to try to sync up, because if I'm spiking out of sync, it's not going to have any effect. And So then what would cause, so how now do I get all these guys to fire faster or slower based, to encode speed or direction or whatever, right? And that's an interesting question. You just pointed out you can manipulate these things physically to change that rate, right? But I don't know, it just seems like
it's been shown that like there are theta cells that when you depolarize
theta
them,
cells?
so cells that have an intrinsic subthreshold theta oscillation, so if you call it the somatic
Subject to theta resonance or oscillation?
actual intrinsic membrane potential oscillation, right? These cells exist, they're called theta cells.
Okay, I just asked about that, and you said, and I said we shouldn't assume that they're self.
Well, but I was talking about like these dendrites. I'm not talking about somatic effects. So I'm talking about a specific class of cells that are called theta cells, which some people have been hunting for using different methods, right? And there's certain brain areas where we have found those kinds of cells.
This is really reliable. People know this, that they found cells that all on their own oscillate at theta.
Yes, subthresholds. There's a subthreshold oscillation.
Yeah.
A subthreshold oscillation in the theta range, which doesn't make the neuron rhythmically fire, but of course the interesting thing is that when you add a current to these neurons, that oscillation can speed up. So that would give you exactly, that's why I mentioned the well-dated paper from 2011. I talked about that, the well-dated 2011 paper, which showed exactly these properties, that they were looking for cells that have an intrinsic...
You reviewed that paper?
Yes, I talked about it right here, and I showed if you remember the VCO law, I talked about the fact, and then there's that they specifically went out to find cells that have a theta oscillation that speeds up with movement linearly, and that has a cosine directional tuning, and have reliable intrinsic membrane potential oscillations, right?
Yes, I just didn't understand it enough at the time.
Right.
Okay.
That was important because these are the input cells to a hybrid or an oscillatory interference model.
Yeah.
I'm very curious about them.
And those cells exist where?
Anterior thalamus, but also lots of other areas. So that they found, was it parahippocampal gyrus,
anterior thalamus,
cortex,
anterior thalamus,
and anterior thalamus.
How would I locate that? I think of thalamus in terms of not the body directions, but the different loci, the different modules in them. So where would those be? Are those do they interspersed with the relay cells, or a different part of the thalamus, or what is it?
I'm not sure.
Let me see if I have that in my notes.
Right.
Anterior thalamus.
We're talking about cells with a theta oscillation in thalamus.
The intrinsic membrane subthreshold oscillations in thalamus.
You can also look up Welday et al. 2011. That's very specifically like those.
What was that one?
Oh, that's the part, that's thalamic nuclei that project to the hippocampal areas. So it's like LGN projects to V1. The anterior nuclei project to the cerviculum.
Which is why they're potential candidates for
inputs.
Reunions, those areas.
Does that mean that they looked elsewhere in the thalamus and didn't find them, or could it be that we see those cells throughout the thalamus?
They listed candidate areas. Again, I don't know exactly. I would just look up the Welday et al. paper.
How do you spell that?
WEL.
Well, just like it sounds.
Right, just like it sounds. Welday.
Oh no, Welday is one word.
2011.
Okay.
They specifically showed these theta cells and recorded, and they showed that particularly the ones in anterior thalamus actually obey the VCO law, so they have a theta oscillation.
I guess the question is really, okay, so these guys are there, are they everywhere throughout the thalamus? Is this a property of thalamus in general for all the different relay centers, or is it just specifically for the projections to the hippocampus?
That is a good anatomical question. I don't think they investigated that.
They must have said something in their techniques about how they chose to look there, or if they only looked one place, or...
I mean, if it's important, I could find out. I can ask Carmen.
She would know
this really well.
Know that really quickly.
Yeah, because she's really into the oscillations.
Well, it'd be nice to know, or I could just look at that paper. I don't know if I want to bother, but if you don't mind,
So this is like 8 hertz, and then when you go in the right direction, it goes up to 8.5 hertz.
You know, because
Wrong
we've
direction,
mapped
it goes down
out
to
the
7.5
anatomy of
hertz.
the cortical relay areas quite a bit. And we've gone through this in detail with other scientists about this. And so I have sort of a mental model or picture in my head about the different cells and how they're connected and so on. And the question is, which of those cells, if any, have this property, which would be sort of, is it the relay cells? Is it the matrix cells? Is it things in TRN? I mean,
That would be a really good question. They don't further specify that in the Weather paper, as far as I remember.
that's key to the whole thing, I think.
Yeah, if you want to understand that circuit, and particularly if we want to use them as more general inputs to
Yeah, we think this is a general property. You know, we talked last time about the idea that perhaps cortex is unique in the fact that it has one of the things that makes it different than the older hippocampal regions is that the evolution had discovered that you needed a way of adjusting time. And therefore you need a centralized timing signal where you don't, and that's scaling, it's timing and scaling. But you may not need that in the hippocampus because if you're mapping out environments that you generally don't have to do with scaling. But if you're trying to see things from a distance or you're trying to hear melodies or whatever, you need that scaling. And so this is like maybe one of the key reasons we have the thalamus at all, is just to do this. So that understanding that's the big idea.
Right, and those are exactly papers that I'm hunting down right now, as I mentioned during stand-up.
So what's the specific question I would ask her?
I guess if these cells that have intrinsic theta oscillation in the substratial membrane oscillations, do they exist throughout the thalamus in all areas? In which particular cells do that?
Okay.
How generic is that phenomenon throughout the thalamus?
Well, there's different cell types in the thalamus. I wouldn't imagine they all have that, but maybe they do. I imagine it might be one classic subset that does it everywhere. I mean, again, I've always thought about the matrix cells as the timing signal. And so it would be very pleasing to find out that that's the ones that do it. But who knows? Maybe they all do it. Maybe none of them. Maybe they don't do it. Who knows? But that would be a piece of confirming, at least supporting evidence, not confirming, but supporting evidence for that hypothesis I've been living with for years. And to know that that's how they work is very interesting. Something I thought other ways about it.
So about oscillations, there's pretty simple ways to create oscillations, right? And
what you need is a voltage-dependent current.
Yeah,
that does it.
But I mean, are these oscillations? Are we thinking about topology included in the methods? Because like the paper that I've been reading about emergent grid cells doesn't necessarily talk too much about the topology of the structure, you know. But if you create a topological simulation, which it's really easy with the natural topology to apply an oscillation through this space just from a random, beginning place. Just by sort of doing some type of topological mean around to a decay function, a decay or a degradation over time, or what do they call it? When the firing rate just goes down automatically over time. And if you add that and then just use a topological mean around that point to reactivate it or to increase the firing rate depending on that, then you get natural oscillations.
Yeah, I think there's a frick ton of ways to get oscillations.
Yeah.
And what I'm trying to hone down on is, what are the specific mechanisms with regard to grid cells, and to what extent do those generalize to cortical grid cells that are used outside of navigation, so are not necessarily medial-entorhinal cortex as targets, but not necessarily conjunct with head direction cells, but wider cortex inputs.
Yeah.
Because those targets of those cells are then putative cortical grid cell, so that it goes towards chasing after this whole hypothesis, which I would like to do.
Okay.
So yeah, I hope everybody's a little bit less confused about LFPs now.
Are we done?
Yeah, I think we're done.
That's good.
Yeah.
That was good.
Yeah.
Anyone else? Thank you. All right.
Okay. Ending the stream right now. Thanks for watching.